Okay, well, I want to thank the organizers for inviting me to participate in this symposium and to tell you a little bit about what my own lab is doing. I thought I would also say a couple of words, okay, thanks, about the Institute for Genomics and Systems Biology where I work. It's a, sort of a unique thing in Chicago right now, a cross-disciplinary institute that is composed of faculty, actually several of which are at Northwestern, many of which are at University of Chicago. Something like 65 faculty are participating in this as fellows, and they come from all sorts of different backgrounds: physics, chemistry, and of course biology, statistics, mathematics. And some of the investigators actually are cross-fertilizing between this project, which is another center in Chicago, which I think is very relevant for the center that is sponsoring the symposium today. This is one of 10 systems biology centers sponsored by the NIH around the country. And ours happens to be the only one that's focused exclusively, largely exclusively at this point, on transcriptional regulatory networks. And I think since there's so much focus on regulation of chromosome and chromatin structure and nucleosomes and so forth in the oncology center here, that there are and actually John Crispino, for example, is a member of both centers. I think there are some really interesting opportunities for cross-fertilization, and John and I talked about one over lunch. But I'm sure there are many. And I would encourage you to, there are seminars and there are monthly group meetings that people at Northwestern can participate in. We have conference calls, and so you're welcome for the grad students and postdocs and even the PIs who aren't too busy to participate in the things going on with that center as well. I think there are probably a lot of things that could be done jointly between the centers. So one of the things we're big on is trying to define large-scale networks, so-called hairballs that Marcia introduced a little earlier, by taking lots of different types of data that have been collected on a somewhat grand scale and integrating them and then culling down to individual genes and proteins or pathways that we think might be important for diagnosis, progression, or therapeutic targets for disease. That's a big, it's actually a big activity of all these faculty and their labs that are participating, including Marcia in IGSB. I also thought I would mention another project that I think that is going on in the Institute that I think dovetails quite nicely with the projects here. And that is, we have two grants, one for mod ENCODE and one for human ENCODE. ENCODE is a project, it's a multi-institutional project sponsored by the NHGRI, the Human Genome Research Institute, and it has the modest goal of completely annotating comprehensively the human genome and the genomes of model organisms. And so one of the ways that we've been going about trying to do these annotations is, in particular, my group is interested and has been funded to annotate cis-regulatory elements and cis-control elements, and I think, again, that overlaps very nicely, is through CHIP and through mapping lots of different proteins that are associated with chromatin. This is just a recent download and clustergram of a bunch of different proteins that we mapped in Drosophila, including things associated with, and this is done across development. We have over 300 data sets on histone deacetylase binding, on methyltransferase binding, on CBP and cofactors, on insulators. We've mapped over 50 transcription factors multiple times in development, and we and others have been doing the same kind of a project on the human genome. And one of the things that we've discovered, or maybe not so surprising, is there are a lot of commonalities between the human genome and the model organism's genome. For example, we find these intriguing regions of the genome that seem to be reservoirs of transcription factors. And we don't know much about them other than they appear to be associated with nucleosome depleted areas. They don't tend to be associated with enhancers. They don't tend to be associated with gene-rich regions or anything like that, but nucleosome occupancy is something that we found. So I think these data sets, which are publicly available, are great data sets to compare the types of data that you all in the Oncology Center are producing that we've heard about earlier today. So, so now I'm going to tell you a couple of, so that's my spiel sort of calling for even more cross-fertilization than is already going on. But what I'm going to tell you about is a couple of examples today of how we've been using these integrated approaches to looking at large-scale data sets to try to narrow down to individual proteins and pathways that are predictive of disease outcomes or to identify new genes involved in cancers. So where I thought I would start is with a story from Drosophila. This is the polytene chromosomes of a Drosophila salivary gland. We had a great discussion over lunch about ideas of collaborating between the two centers to try to get at the mechanism by which puffs form. These are decondensing regions that are associated with transcription in these polytene chromosomes. But maybe we'll be able to talk about that next year. This year, I'm going to tell you a story about how we've been assaying protein interactions and gene expression patterns and transcription factor binding sites in this genome, to try to put together larger scale networks and pathways and have already been elucidated in very common developmental biology areas. And one of those is the so-called segmentation regulatory network. This network is probably the most heavily studied developmental biology example of how transcriptional regulation specifies the body plan in any organism. And among other notables, the initial identification of many of these genes led to the Nobel Prize for Nusslein Vohard and Eric Wieschaus about 15 years ago. And before and since then, there's been an enormous amount done to lead to understanding of how these, almost all of which are transcription factors in these first three steps of the hierarchy, are working together to control this process of setting up the anterior-posterior axis in the embryo, leading to segmentation in these organisms. And what's most interesting for the purpose of today's talk is that at the end of this cascade, where these maternally deposited transcriptional factors activate a set of so-called gap genes, which are called that because when you mutate them, you get gaps in the cuticle and the embryo, and these transcription factors activate these transcription factors. Well, these transcription factors then go on and activate a whole bunch of pathways that are highly conserved between humans and flies and are almost all implicated in human cancers. These are things like the hedgehog pathway, the Wnt signaling, the TGF-beta signaling, and so forth. So what I'm going to tell you about today is how we've been building out a transcriptional regulatory network for some time now around this level of the hierarchy to look for new targets in these regulatory pathways. And I'll give you an example of one that has been quite intriguing to us. So I'm going to restrict my talk today to Fushi-Chiratsu and even skipped. These are two core pair rule genes that are expressed. This is their actual pattern of expression. It's not a cartoon. It's a microscopy image of an embryo expressing Fushi-Chiratsu and even skipped, both homeodomain transcriptional factors. And then this is the segmental pattern that results after these factors specify in these epithelial cells. This is stage 5 cellular blastoderm for the Drosophila aficionados after they specify their targets. So we mapped FUTs and we mapped EVE binding sites throughout the genome, and simultaneously, some years ago, we developed a little method for extracting the mutant embryos, because you can't tell at stage 5 when they all look the same. They haven't. There's no phenotypic difference between them. To molecularly distinguish the mutants from the wild types. And then we did time courses of the mutant EVE, the mutant FUTs, and compared them to wild type and identified genes that required EVE for upregulation. These would be like the blue guys here. And genes that required EVE for downregulation. These are the ones that are upregulated in the mutants. And these are binding sites nearby these regulated genes. And so this is a core set of several hundred genes that we think are most likely to be direct targets of these two factors. Simultaneously, while that initial work was going on, there was a publication, now this was seven years ago, that came out looking at the large-scale yeast-2 hybrid interaction data set for Drosophila proteins. Close to 80% of open reading frames were assayed over 22,000 interactions. Actually, not until very recently, like now, and it's still not published, has this been updated with IP mass spec kind of project from Spiros Artavanis lab. And so we did this initial work with this particular network. We developed a training set based on a variety of parameters that you can read here to give them scores between 0 to 1, and then we validated those scores and showed that the ones that were closer to 1 were much more likely to be found in the literature than the ones that were closer to 0. And so we had an independent validation of these scores. And then this is my only sort of semi-quantitative slide. Then what we did is we built a network. And there's a lot of different ways to build networks. This one we published now 6 years ago. It still works. We've done a lot of modifications to it, but the basic idea is the same, which is you lay down a set of possible interactions, like this protein binds this target gene, or this protein binds to this other protein. And each of those bind terms is an edge in the network, and each of the protein or gene terms is a node in the network, and you get a huge sort of hairball that looks like this. And the hairball is actually weighted, and it's weighted by a probability matrix where we develop a joint probability. The simplest one would be to say take the likelihood based on this 0 to 1 of a yeast-2 hybrid and take some other statistic like gene expression association or probability based on your ChIP-seq data that a particular gene is a target of a particular protein, and you can add those all up into a joint probability. And then you lay down the network, you remove an edge, you recalculate it, you move a node, you recalculate it, and you do that in a big computer millions and millions of times, and you come up with a bunch of nodes and edges that actually have probabilities underneath them, which is somewhat useful because it helps you to narrow down taking only the highest probability or looking at other properties in the network to sets of proteins or genes that you might be interested in. One that struck our fancy was the CG9924. What was notable about it was how interconnected it is. So you can see it's a hub in this network. It's interacting. It's both a target of Eve and FUTs transcriptionally. It's interacting with a lot of other proteins directly. It's interacting with the Jun kinase signaling pathway in this sort of computationally derived network. In fact, if you look at CG9924, what you see is that if you try to go from any one point in the network to any other point in the network, it's the node that you have to pass through most often. It's the most highly interconnected protein in this network, and we were interested in it furthermore because it hadn't been really deeply studied in Drosophila. In fact, there were no papers on it in Drosophila when we started the research. There was one paper in human, and this thing is called SPOP. We call it Drosophila SPOP. It's associated with sarcoma autoantigen in a paper we couldn't read because it was in Japanese. But we at least were able to get the translated abstract. And so that was the only reference to it. And so we made antibodies. It actually is located in the segmental grooves during the process of segmentation. And the mutants look like this. And another group about the same time as our first stuff came out on this, published the mutant, and they call it roadkill because it looks kind of like a hedgehog or something that's been run over. One of the things that's notable about it, though, is it does have segmental defects, however obscured they might be in this mess. And so we validated in vitro that it can interact with Jun kinase phosphatase, which is also known as puckered in Drosophila. And then instead of going in the embryo where we have this really kind of messy phenotype, we decided to turn to the Drosophila eye, where you can overexpress TNF. In flies it's called Eiger, but you can overexpress TNF. This is an expression where TNF is sort of lightly expressed. So if you activate that expression, you'll get apoptosis. Here's it's really strongly overexpressed. So tumor necrosis factor is overexpressed, and you actually push the cells over into apoptosis Z cells. So what we wondered is, does this interaction with puckered, is it negative, thereby acting as a negative regulator of a negative regulator of Jun kinase, and thereby activating the pathway, or is it positive? So we put it in into these genetic backgrounds, and we showed that in fact it rescues this eye phenotype from the overexpression. So this suggests that, again, now we've got a mutant, so we've got triple negative logic, genetic logic going on. I'll move back to biochemistry. And so this suggests that DSPOP is actually a negative regulator of Jun kinase signaling. This was kind of satisfying. Here you can show that in fact puckered, it's downregulated at the protein level. So it's degraded when you add SPOP, and it gets ubiquitinated. When you put a MG132, which is a proteasome inhibitor there, you get hyper ubiquitinated puckered. And this fits with something that I didn't tell you about, which is the protein itself has a BTB domain, and it looks like a member of an adapter complex, and it's got a math domain. It looks like a member of adapter complex between CUL3 ligases and their substrates. And so we sort of wondered, this new member of the Jun kinase signaling pathway, does it have any association with cancer? So we ran a tissue microarray of about 20 different human cancers and asked whether it was overexpressed or underexpressed in association with any type of cancer. And what we found was that it is overexpressed specifically in clear cell renal cell carcinomas, so in kidney cancers. And clear cell renal cell carcinomas are the most common type of kidney cancers. It's almost in 100% of clear cell renal cell carcinomas. at high levels, whereas in normal tissues, it's at 100% negative staining. And so it looks like a nice biomarker for clear cell renal cell carcinoma. So we published this last year in this paper here. And since then, we've been trying to figure out what it might be doing in these kidney cancer cells. So one of the first clues is we looked at clear cell renal cell carcinomas, and we noticed that there was an abundance of the protein in the cytoplasm. In the initial report, it was shown in the nucleus, and in normal tissue, kidney tissue, it's at a low level and it's in the nucleus. In CACI2 cells, which are a clear cell renal cell carcinoma derivative, it's in the cytoplasm, it's shown here. And in HEC293 cells, which human embryonic kidney cells, it's in the nucleus. So it looks like it's, and then what we can do is show that that's true with a tagged version where we take a tagged SPOP, and it's also in the kidney cancer cells, it's in the cytoplasm. It's a little hard to see in this light. And in the normal cells, it's in the nucleus. We can lop off its nuclear localization domain in the HEC293 cells, and it goes to the cytoplasm. So shortly after we made the initial discovery, we contacted Brenda Schulman's group at St. Jude's. And she's an expert at crystallizing, making co-crystals of these type of complexes. We wondered what it looked like and if it actually really would co-crystallize and interact. And so this is a crystal structure that, that Brenda's group produced. And it actually forms this kind of dimer. You can see it's got a domain here that interacts with the substrate and a domain here that interacts with the COL3. So that's what it looks like. We were interested about what its targets might be in the cytoplasm and in the kidney cancer cells. And we knew that it targeted Jun kinase signaling, or ERK signaling, in fly cells. So we wondered whether it might target phosphatases in human cells as well. So we noticed that of the different interacting proteins, here's puckered, but there were a couple more that we found we and others have shown interact. There's a sort of very degenerate consensus sequence. We scan the genome and look for different phosphatases that contain that sequence, and we found USP6, DUSP7, and PTEN. These are all phosphatases, of course, and we showed that they interact in vitro as well. Further, we've shown that they get hyperubiquitinated, as does puckered in the same ubiquitination assay. So probably more importantly, when we look at actual kidney tumors, what we find is that SPOP, again, is overexpressed in the clear cell RCCs, but USP7 and also 6 are expressed at high levels in normal cells and lower levels in clear cell RCC, and same with PTEN. So I told you we made this nuclear localization minus SPOP, and what we found with these cells, these HEC293 cells, and also HeLa cells, if you put it in HeLa cells, that they hyperproliferate relative to control cells, and if you take the full length, they actually undergo some apoptosis. This makes sense because the full length is stimulating apoptosis by acting as a positive regulator of TNF signaling. And this is doing something else, presumably, and we think that one of the things that it may be doing is downregulating PTEN. And this is showing in HeLa cells, if you knock down SPOP, this is also true for HEC cells, if you knock down SPOP, you don't see a major effect. When you knock down SPOP in CACI cells, it actually causes them to undergo apoptosis. And so it seems to be specifically required and also sufficient for this hyperproliferation. Looking in Xenograft model, we recapitulate this result showing that these mice get tumors when you take a HEC293 cell and do a subcutaneous injection. We're actually now, with some collaborators in urology, doing these xenografts onto the renal capsule inside, doing surgery on the mice. But this was the initial result, and the full-length SPOP doesn't have this effect. So I didn't show you the DAX results, but DAX, of course, is an apoptotic stimulator, and it is also, SPOP is also degrading DAX. So our idea is that SPOP, when it's in the nucleus, everything's hunky-dory, and when it's outside the nucleus, it's downregulating these factors that normally tell a cell to apoptose when it goes out of control. And when it's also downregulating P10, which normally is blocking proliferation. Now, what I didn't mention, and this is new data, is of course VHL and HIF are widely recognized to be important genes in controlling kidney cancer. VHL, it's another class like SPOP of these adapters between ligases, ubiquitin ligases, and their substrates. And HIF is one of its substrates. And it turns out that when VHL is mutated, you get kidney cancers, and HIF goes to the nucleus. Well, our latest data suggests that HIF, in fact, is regulating SPOP, is upregulating SPOP. And so it sort of draws these two pathways together, which I think is pretty exciting. That was just deductive reasoning. That wasn't in experiments. It wasn't large-scale network bashing. But certainly the discovery of SPOP was. OK, so in the second part of my talk today, I'm going to actually switch gears and tell you how, sort of encouraged by these results, moving from building out networks in Drosophila based on transcriptional regulatory and protein interactions and so forth to a particular human cancer, we wondered if we take the same approach directly in a cancer or a cancer cell model, whether we might also have some success. And so for a good number of years, my lab has been studying, and actually during my PhD thesis, I studied nuclear receptors in Drosophila. So nuclear receptors, of course, are transcription factors that actually also act as a signaling molecule. So they receive a hormone signal, and then they regulate downstream targets in the genome directly, sometimes indirectly, but the canonical model is directly. And nuclear receptors, of course, are very important in breast cancer, particularly the estrogen receptor, which is associated with breast cancer progression. Breast cancers arise from these epithelial cells, most often from the ducts. And what I'm going to tell you about is how one project where we've used MCF7 cells as a model. We build these networks, identify biomarkers from these networks, and tie those biomarkers to predictive outcome. And then another study where we've been sequencing actually the genomes or the transcriptomes of the primary tumors themselves. And so it's already been alluded to today that by Marcia that you've got these triple-negative breast cancers, and you've got ER-positive breast cancers, and your probability of survival is sort of not too good with triple-negative breast cancer, much better with estrogen receptor positive. And this is in no small part to the fact that you've got selective agonists like tamoxifen that are used to treat these cancers, but also the tumors themselves do have biological differences in their aggressiveness, and as Marcia said, these are much more prone to being metastatic regardless of the treatment. So of course, there are different subtypes that are defined on the molecular basis And these roughly fall into ER positive and triple negative for this red and this blue. And so I'm going to come back to these molecular subtypes that have been defined just on gene expression profiling. So what we wanted to do is take the MCF7 cell line, which is ER positive. We're mapping the estrogen response. You can see here that these are just arbitrary units, but you can see that proliferation goes way up when you add estrogen to these cells. And I mentioned these other facts here. The sort of Cartoon Network version of how we think in very broad strokes is that the estrogen receptor bound to estrogen activates a lot of target genes and represses a lot of target genes that lead to proliferation. Also, it's known to activate c-Myc, everybody's favorite oncogene. And this has been known for a long time. And c-Myc exacerbates this problem as well. So we did ChIP-ChIP, in this case, on c-Myc and ER across the genome. We identified sets of direct or putative direct targets. We also did an RNAi screen, which we've done now across the entire genome, which was a huge undertaking by Ruby Dar and my group. And what she did is she looked at proliferation plus or minus estrogen and identified estrogen-dependent proliferation and also estrogen-independent proliferation. This happens to be just a data set for a bunch of, I thought I would put this here, it's, chromatin factors, chromatin remodeling factors that are across the scale in terms of their influence on the proliferation of the cells. They're all targets of either estrogen receptor, c-Myc, or both. Then we map these on top of networks, and we pull out individual interesting factors. And the one I'm showing here is H2AZ. H2AZ, and maybe you guys can tell me why this is the case. But what we find with H2AZ is it's a direct target of c-Myc. It's upregulated sometime later after estrogen, and c-Myc expression leads to its binding to these E-boxes, and it's upregulated sometime after c-Myc binds. But when we look at the levels of H2AZ in primary tumors, we find that patients with very high levels of H2AZ have very poor prognosis. This is a Kaplan-Meier plot of survival over time. And either low levels or medium levels, they do much better. In fact, these patients are 3 times more likely to have metastatic disease, which is congruent with the idea that they're associated with triple negatives as well. We've done some screening, identified through a collaboration looking at a bunch of HDAC inhibitors, a set of compounds that are actually inhibiting the cells that are highly expressing this at some relatively low levels that could be good leads for further drug development. So that's one story. And but we were actually interested in the idea that maybe more than estrogen receptor could be important in the transcriptional regulatory networks that are leading to disease progression in breast cancer. And ideally what we would do is map all of the nuclear receptors that are expressed in these breast cancer cells. The problem was that ChIP-grade antibodies were at best available for 30%. So what we did is we developed an approach using back recombinering where we put a GFP or another tag into a part of a human chromosome that's been cloned into a bacterial artificial chromosome, and we fuse it in frame, typically to the C terminus, but sometimes to the N terminus of the transcription factor in question. And so we've actually done this for all the nuclear receptors in the human genome, and there are about 24 of them expressed in MCF7 cells. These back recombined transcription factors, when we put them back into, we make stably transgenic cell lines, when we put them back into these cells, they go to the right place, they go to the nucleus. And more importantly, when we map using antibody against endogenous factor, in this case it's FOXA1, but we've done several other ones, or an antibody against the tag, in the tagged version, we get the same binding sites. There's typically 80% overlap when we do this, and that's very similar to the overlap if you do just the endogenous in replicate, given the limitations of the peak calling algorithms and the overlap algorithms and so forth. So we went ahead and we mapped all of these in these cells, and this is where they bind relative to the transcriptional start sites. And this is not the topic of today's talk, but one of those possibilities for overlap between what people are doing in this center and what we're doing. There's this observation that there's some factors like the estrogen receptor that are very distally found from the binding sites, from the transcriptional start sites of the genes that they regulate. There's other factors like here's CREB and ATF. Here's a couple of orphan receptors that are bound right there at the promoter. And so, what is it about these factors, who they interact with, and the chromatin structure that leads to these very different scenarios? But we do see them, and we see a full gamut. This is actually a conservation plot now. You can actually see the darker it is, the more conservative. You can see that there is, if you start looking at the relative conservation of the nucleotides, as you get closer and closer to actual binding site, it becomes higher and higher conserved. So this is good to see. The other thing we can do is we can cluster this data. So we're now clustering not gene expression, but we're clustering binding sites throughout the genome. And here you have a cluster where you've got the estrogen receptor, you've got FOXA1, which is a known interactor, but you also have these retinoic acid receptors. And I'm going to talk a little bit about what we've done with these. There's a lot of very interesting stuff in here that I'm not going to talk about. So this is, if you don't like clustergrams, this is what it looks like in a Venn diagram. Actually, 70% approximately of estrogen receptor targets overlap with RAR alpha or RAR gamma. It's a side note. This tagging approach allows you to distinguish between different isoforms from the same locus or different isoforms from paralogous loci in the genome very nicely. In this particular case, RAR alpha and gamma look like they may be largely functionally redundant in terms of their binding. Nonetheless, what we wanted to do is transcriptional profiling in the presence of ligands against a pan-RAR agonist, ATRA, or selective agonists against RAR alpha or RAR gamma, and ask the question, which of these binding sites are differentially regulated in the presence of hormone. We also did this with estrogen and made the sort of stunning, I think, inverse correlative observation that genes that are upregulated by estrogen typically are downregulated by retinoic acids, and vice versa. Now, there's examples of synergy where they're working together, but overall, something like 70 or 80% of the genes look like they have opposite effects. And these are all genes that have RAR alpha, gamma, or estrogen receptor bound. So I don't have time to go into more detail, but we have at least one biochemical explanation of how our retinoic acid receptors and estrogen receptors seem to be competing for the same or very nearby sites in the genome. But in the spirit of the theme of the talk, I wanted to show you another example where instead of using one protein or one pathway, what we do to look for sets of signatures of biomarkers that can have a predictability of patient outcome is here are over 300 tumors that have been gene expression profiled, breast cancers that have been gene expression profiled. They fall into these different categories that we talked about earlier. And then these plots here are the relative density of retinoic acid receptor and estrogen binding sites nearby those genes. So now if we filter on that density, we arrive at a different set of genes, and you set the threshold at different levels, but we set at a level that maybe takes out 30% of the genes, leaving 70% back in the cluster. And then we look at the combined metric of how regulated those genes are by these factors. And we use that metric to do another Kaplan-Meier plot. And you can see that depending on and then we just separate into things that are highly regulated, not regulated, and something in between. What we find are factors or sets of genes that are upregulated by the estrogen receptor are actually associated with poor prognosis, and ones that are upregulated by the retinoic acid receptor with better prognosis, and vice versa. This is consistent with some studies that people have done in vitro, at least, showing that retinoic acids can induce these cells to undergo cell death versus estrogens, which have the opposite effect. This is also true for relapse-free survival. So, so far I've told you about how you can improve predictability using individual biomarkers, combinations of biomarkers, but ultimately, big challenge in the field, I think, one of the big challenges is uniting the signaling kind of networks that we've heard about earlier with the transcriptional kind of networks that I've been talking about, and connecting those to the genetic state of the cells, because ultimately a large part of the cancer phenotype is due to mutations, right? And so what we've done to take a first little step in that direction is we've started to do transcriptome sequencing of breast cancer. We're also doing exome sequencing. But the study I'm going to tell you about right now, we did 11 ER positive and 15 triple negative breast cancers to about 1 gigabit bytes per genome. When you look at just counting the transcripts and you do clustering, what you find is that now in red are triple negative, in blue are ER positive, open circles are Caucasians, closed circles are African Americans. These are patients from the University of Chicago. We find, as expected, that you get a cluster over here. This is a principal component analysis. Cluster over here of the triple negatives, a cluster over here with the ER positives. So this is the expected result. It's just another way of clustering. One of the things you can do with transcriptome sequencing is you get a very good bead on isoforms. Like you get a very good idea of which isoforms are expressed and to what levels in the different tumor types. And you can see here are just 5 different genes. Doesn't matter what they are. What's important is that in many of the cases, there's one isoform that's not expressed in ER positives or triple negatives, and other isoforms are having sort of the inverse relationship. And it turns out if you just look at isoform data, you can do this clustering as well. And so just using isoforms, whether or not an isoform is expressed at appreciable levels in a tumor, and you can generate a panel of a few dozen of these, and you can be very predictive about which subtype of cancer that you're looking at. But this is all expression, and what we'd like to do is use this information to actually identify nucleotide variants or indels that are associated with the disease. The big issue here is can you find genetic signatures? If you could find genetic signatures, maybe we wouldn't have to worry so much about these more complicated signatures. Maybe we will. So, but here's what we found. In this data set, we got about 35,000 single nucleotide variants. We don't know whether they're, at this point, variants that are somatically derived or whether they're inherited. And then we look at where they fall within the gene. We look at their allele frequency. Are they rare versus common? We can use the 1,000 Genomes Project, the HapMap project, the SNPDB, all these various papers that have come out on exome sequencing to identify rare versus common alleles. If it hasn't been seen before, we try to be very stringent. If it hasn't been seen before in any of these databases, we call it rare. If it has been seen even once, we call it common. And so then we use evolutionary constraint. And why we do that, you can imagine that if you have a highly conserved nucleotide throughout the 30 or so vertebrate species that have been sequenced, then that highly conserved nucleotide, when it's mutated, would be deleterious. And so we're looking for things that are more deleterious versus less deleterious. And so one can assign a score on how evolutionarily conserved they are, the so-called Gert Score. So things with high Gert scores are more deleterious. Things with low scores are neutral. You can see that the common alleles follow a distribution that is quite different than the distribution that the rare alleles show. The rare alleles tend to be more deleterious. And so we're interested in these rare deleterious alleles. Now, if we now cluster not on gene expression, but on genetic sequence, on DNA sequence, we're clustering on variants. If we take the common variants, what we do is we cluster based on race. So these are your Caucasians, here are your African Americans. Here's a couple of self-reported Caucasians. We're not sure what, they don't fit with either group, but in any case, we're clustering based on race, and this is what one might expect. This is why GWAS studies are so difficult, because they're looking at these common variants and always having to deal with controlling for population stratification. However, if we look at these rare deleterious variants, we get a much nicer separation out of the ER positive into triple negatives. So that means if we filter for the rare deleterious alleles, we're able to create genetic signatures that are predictive of who's going to be ER positive and who's going to be triple negative. We wondered, are these germline or are these somatic? So we verified by sequenome now more than 113, but a bunch of them. And we looked at adjacent tissue versus the tumor, and we found that almost, the vast majority, and this 98%, are germline. So these are inherited alleles. We wanted to sort of formally show that this is statistically significant, these differences between these different populations. So what we did, we ran simulations. This is if you just randomly assign these alleles. There's about 300 of them that we're using. And so we randomly assign them to triple negative or ER positive. And this is a FST, or the F statistic. This is a statistic testing for population differences. And this is the observed at a sort of a cutoff, I think, of a Gert score of 4, and this is a cutoff of a Gert score of 5. So these are extremely significant. You never observe in more than 10,000 simulations. This is now these simulations done at multiple different time points, and in the closed circles are the common variants, and also we're looking at multiple different Gert scores, okay? So sorry, I didn't mean time points, I meant Gert scores. And this is the FST values. You can see you get this progressive and then shooting up at the high Gert scores of the rare alleles, being informative in terms of distinguishing these populations from one another. These in red are the actual rare variants. In the columns are the patients, and these are the genes in the rows. And you can see they fall into known pathways. So we wanted to replicate this work. So what we did, and I'm almost done. What we did is we took 50 patients, actually from Toronto, and we looked at 60 single nucleotide variants. And this is a discovery set. This is true positive versus false positive rate. This is a discovery set in red. This is a validation set in blue. What I didn't show is a control set of 50 women that didn't have breast cancer. They're right along this line. These are pretty good numbers, actually. We think they're going to improve as we get more variance in larger population sizes. But if you look at, say, the 50% mark here, what this is saying for about 80 or 90%, or for 50% of the patients, and we know which ones, we can look at their inherited variation. And if they get breast cancer, we're 80 or 90% accurate at determining which type of breast cancer they're going to get. And so you can imagine this could be extremely useful in terms of getting your genotype done and then knowing what your risk level is and whether you should get mammograms at earlier age, or if you do see something in the mammogram, how aggressively it should be treated. So just to conclude from this part, isoform expression can be used to stratify disease subtypes. It's neat. Breast cancer is associated with large numbers of rare inherited, what we call RID alleles. RID alleles can be used to stratify disease subtypes. Breast cancer is a complex trait and has a quantitative genetic basis. It means these alleles are working together. I didn't show you, we ran an experiment where we looked at 150 randomly selected alleles in lots of different combinations, and any 150 would do. So this suggests that each one of these is giving some kind of contribution. It's just not a few drivers. Yet rare alleles seem to be driving this disease. Common polymorphisms likely have little contribution. So why then, you might ask, what about the GWAS studies? Why didn't they show more than they did? We would argue that linkage equilibrium, along with rare variants contributing to common disease, makes them a really uphill battle. That's saying that if you've got something rare, then the chances that it's associated with any given common variation, it only happens rarely, so you have to sample massive numbers of individuals. And then if there's any linkage disequilibrium, or any linkage equilibrium at all, recombination moves it out of there. And so it's actually a little black box in the realized heritability equation in all the GWAS studies that they don't know the value of. And so our speculation is that probably other cancers and other human diseases obey similar fundamental rules. I'll end there. I just want to say that we, as part of an initiative that actually people both at the University of Chicago and Northwestern are participating in, where we've really scaled up our tumor sequencing. We've sequenced over 200 tumors by now. This is just a subset of them. And this is a cluster of rare deleterious variants now among a whole bunch of them. One of the things I want to show you that has come out of this sort of global clustering thus far is that here's ER positives down here on the sort of global clustergram. Here's triple negatives way up here. These are head and neck cancers. So there's subsets of head and neck cancers that are more like triple negative breast cancer than they are like other head and neck cancers, and vice versa. And so these genetic signatures are potentially a much more reliable or powerful way of lens to look at things than sort of traditional clinical and anatomical pathology. And just to reiterate, we're one little step closer to understanding how genetic state might be able to lead to predictions. A lot of people are working in this area as well and unifying these two areas. How do these changes result in variation in these complex networks is going to be a really fun and exciting thing to engage in, and I think that goes from signaling to transcriptional regulation to chromatin state to genetic lesions, and sort of there's a lot to do in a lot of areas to cross-pollinate. So Casey, Megan, Tom, and these collaborators here, and Mark, who's our director of the Genome Analysis Core, responsible for the sequencing. The blue folks are the SPOP story on kidney cancer, and the green folks are the stories on the nuclear receptor networks in breast cancer. I'll end there. Thank you.